All engine running.
Absolute genius.
Get this.
Welcome.
This is the show where we bring you
Science.
What that essentially means is
discoveries,
Advances.
Questions. Research.
technology.
Unbelievable.
Without further ado,
This is The Naked Scientists. Hello, welcome to the Naked Scientists podcast. This is the show that brings you the biggest breakthroughs and also talks to the major movers and shakers in the worlds of science, technology and medicine. I'm Chris Smith, and this week we're looking at the elements of life frozen deep in time. Across our planet, natural archives preserve the biological footprints of species long gone, from woolly mammoths at the macro scale through plants and seeds to dormant bacteria and viruses at the micro end of the spectrum. And one environment that safeguards some of this material in the best condition of all is the cold. In other words, in ice. So this week, we're going to look at what's sitting in nature's deep freeze and some amazing stories of what we can actually bring back to life, in some cases, thousands of years later. Let's begin with the largest. In 2010, tusk hunters in the Arctic Circle discovered the perfectly intact body of a young woolly mammoth that had been preserved in the permafrost. They called it Yucca, after the nearby village of Yukagir, where it was found. The trunk, the fur, and even the brain are all beautifully preserved, and so too is its biochemistry, because DNA and even traces of notoriously fragile RNA molecules, the recipes for the proteins being made in the animal's cells, can still be recovered from the 40,000-year-old tissue. So the tantalising question is, could the genetic information inside specimens like this be used to bring them back from extinction? Ben Lamb at Colossal Laboratories and Biosciences in Boston is aiming to do just that. Armed with the genetic codes of a host of mammoths like Yucca, they've identified the key differences that set them apart from their close elephant relatives, and they're now in the process of engineering those genetic changes back into elephant DNA so they can ultimately use cloning techniques to bring these animals back to life.
So mammoths appeared a little over 1 million years ago in the evolutionary tree. They went extinct about 4,000 years ago. So what's crazy, most people don't realize this, is that mammoths were alive at the same time that the Great Pyramids of Giza were standing, which is amazing, right? When you think about that in perspective. They were a cold-adapted elephant lineage, so they were pachyderms. They lived mostly in the mammoth steppe ecosystem, but had distribution and migratory patterns. So think of like northern Siberia, Alaska, Russia, Canada, but then they came all the way down even into the central parts of America because they traversed very wide different places. They were herbivores, so they, mostly grasses and trees and shrubs, and they were absolutely critical to the ecosystem with dispersing seeds and defecation across and helping make that ecosystem really flourish at the time.
A big African elephant weighs as much as a bus. Were mammoths bigger?
They were about the size of African elephants today, a little bit bigger than Asian elephants, but somewhere between Asian elephants and African elephants. Sometimes people think they were much, much larger, but they weren't.
And where did they go? Why are they no longer with us?
So they went extinct for many of reasons, right? Changing climates, the introduction of anthropologic effects from early man. You can almost track on every single continent the introduction of early man and the decline of megafauna. There were some cataclysmic events that occurred, in the late stages of the Ice Age that we think also probably had some role in their demise. And then the last ones actually died most likely, it's a sad story, due to inbreeding on Wrangel Island, as well as loss of water because they were on an isolated island. It is a little bit of a tragic story, but if you look at their migratory patterns and when they started to go extinct in the last part of their lineage, most of them were driven further and further north into isolated island pockets.
Your plan is to what? Try to de-extinctify, resurrect these animals, or is it to extract information that we still have about them and just learn about them?
No, it's to bring them back, just like we did with our dire wolves, and we're working on other species like the dodo and whatnot. We want to identify the core genes that really made a mammoth, and bring those back and engineer them into their closest living relative, being the Asian elephant, which is our genetic donor, and actually have mammoths walking around the planet again that have all of the core phenotypes that made mammoths unique and uniquely suited for their ecosystem.
And what have we got as our starting material? What have we got to work with in pursuit of this?
So we actually have frozen samples and materials from mammoths that have passed away that died in the permafrost, that got buried and frozen over time. And what Colossal's been able to do is take about 104 mammoth samples from these frozen remains that go across 1.3 million years. So we've got 104 samples from different unique mammoths that lived and existed over a million plus years of time.
What's the condition of the material? If you had some of my tissue, you could easily read the genome because the DNA would hopefully be all intact and very readable. What's the state of these frozen samples?
Ancient DNA and DNA as a whole degrades very, very quickly. Optimal conditions for storing them are cold, dry, dark places, right? And so that's one of the things that was great about the mammoths is that they died in cold, dry places, so they got well preserved. But even their DNA is massively fragmented. So we actually have to spend a lot of time and use artificial intelligence to do what's called ancestral state reconstruction models. So we actually have to use AI to actually rebuild the genomes and compare it to other known elephant genomes, some of which we've also created. And that's why you need so much DNA over such a long period of time so that you can build a synthetic genome from all of these individual genomes.
There are about 3 billion letters in a human genome. There's probably going to be something similar, I'd guess, in running a mammoth. But I couldn't, with the best will in the world, make a human tomorrow just from what we know from the Human Genome Project. So how can you turn that genetic map, that DNA sequence, back into a living, breathing, walking woolly mammoth?
Well, Asian elephants are about 99.7% the same genetically in terms of related to a mammoth. And so we focus on the parts of the genome of those 104 mammoth genomes across that 1.3 million years, and they actually stayed the same over a vast amount of time. And those are the genes that gave rise to the core physical attributes that made them woolly with small ears and additional fat layers so that they could survive some of the coldest temperatures during the Pleistocene. And so we really spend a lot of time identifying those genes and then engineering those genes on to the Asian elephant platform because all the other stuff's roughly the same.
Right, so by comparing what the elephant that we have today has and what the mammoth does that's different, you can then say, well, I'll put the different bits and superimpose those in the elephant and replace the bits in the elephant. So we should end up with a mammoth genome. But that still doesn't get us a real-life mammoth, does it? So what's involved in turning that genome into a living, breathing mammoth?
Most people have heard of the project from the 1990s where they actually cloned a sheep named Dolly. That process is called somatic cell nuclear transfer. And that's where we take a somatic cell, we look at the nucleus, we take the nucleus out of the somatic cell, put it in that of an egg cell, we stimulate that basically creates an embryo that can be implanted into a surrogate.
We know this works with mice. We know it works with Dolly the sheep. We've had Ian Wilmot, now sadly not with us, who cloned Dolly the sheep on this show.
Oh,
but
that's amazing.
It gets harder and harder the more complicated the species is. Has anyone ever cloned an elephant?
We will be the first to ever clone an elephant. Only about 18 species have been cloned, and that's not a function of technical depth, that's a function of focus and funding.
So how far along that pathway are you? Have you edited all the mammoth changes onto the elephant sequence yet, or is this a work in progress?
We are in the editing process, right? And so obviously, in addition to creating our woolly mice last year, which were a testament to the technologies in platform, we've edited over 25 different genes into that of the Asian elephant. So that's multiple edits in the actual genomes that we're going to be using from our genetic donor. So we've already done that process. We've decided that originally there were about 65 targets. Those are not genes, those are individual edits in those genomes. We've now surpassed that. It'll be probably more like 85 to 90 edits in our first mammoths.
Now, the final step in doing this is going to be to take that egg that's ready to go, the elephant egg cell with the edited mammoth DNA in it and put that back into an elephant as the surrogate. Does IVF like that work in an elephant?
So I don't know if we'll be the first. We're working with Dr. Thomas Hildebrandt, who is leading the genetic rescue for the northern white rhino, which we're also involved in saving that species. And so a lot of those same technologies that we're co-developing have applications to elephants, and we're starting that process of trials with non-reproductively viable females. So we're working with females that couldn't be pregnant so that we can help them get pregnant. That process is in the process of starting. And so it'll have massive applications to elephant conservation when it works. But just like no one had done the same thing in wolves before us, no one's going to be doing the same thing in some of the other species that we're working on as well. So the assisted reproductive technologies for non-model species, like some of our genome engineering, is, world-breaking science.
Certainly sounds like it. And you're probably, after that interview, a bit like me, surprised at quite how far that research has come already. Thanks very much to Ben Lamb at Colossal Laboratories and Biosciences in Boston.
The Naked Scientists podcast is produced in association with Spitfire, cost-effective voice, internet and IP engineering services for UK businesses. Find out how Spitfire can empower your company at spitfire.co.uk.
Music in the programme is sponsored by Epidemic Sound, perfect music for audio and video productions.
This is the Naked Scientist podcast with me, Chris Smith, and today we're opening nature's freezer to find out what is lurking in the permafrost. Coming up, we'll examine bacteria and their remarkable ability to survive inside Norway's glaciers. But first, to plants. These too sometimes turn up trapped in permafrost, either as seeds or just plant tissue, and scientists have managed to bring them back to life too. One celebrated example is a campion found frozen for 40,000 years inside a Siberian squirrel burrow. And seeds recovered from archaeological digs going back millennia have also been successfully germinated. Jen Bromley is a researcher in plant sciences at Churchill College, Cambridge.
Well, different plants take a different approach to life in the same way that many animals, many microbes take different approaches to life. So every different organism has a different sort of strategy in order to maintain its genetic presence in the environment. So if you think about how organisms, what our end goal is, it's about maintaining genetic presence. And so we reproduce to maintain genetic presence. And some plants will do that quickly. So you have like annual plants. They literally are one and done in a year. But the next year, you'll see their seeds come up again. And that's genetic presence that's being maintained.
The other way that some plants do persist is that they set seeds, but then those seeds don't immediately come up. They're like a time capsule waiting to burst open and bring the plant back into existence for an ill-defined length of time. We had Sarah Salon on this programme from Israel, who famously went to archaeological digs and found seeds, including date pips spat out by people in the Middle East 2,000-3,000 years ago. She managed to get those to come back to life. So it's extraordinary. I mean, she says she has one of them growing in her garden or in her lab. But it's extraordinary that these seeds can persist and remain viable for thousands of years.
Seeds are like a little pool of dormant genetic material that can persist, and it can wait until the conditions are right for it to germinate. And hearing about Sarah's work and knowing that, some of these seeds were 2,000 to 3,000 years old, it gives me a bit of happiness and positivity that the seeds in my seed store, in my shed, which are a few years old, will probably germinate.
Are there seeds that have gone longer than a few thousand years ago? Because, we've heard about mammoths where their DNA has remained intact in ice. Given that seeds are already made to last, are there examples of environments where seeds have been set, found themselves locked away in places like ice, for example, and they have now sort of been in suspended animation since?
About 2012, a paper came out about the narrow-leaved campion, or Silene stennifolia. And in that, they had found 30,000-year-old seeds from the Siberian permafrost, and they were encapsulated within the fruit that squirrels burrowed and left in their stores. These seeds got locked away in the permafrost and were only found relatively recently now. So they've been there for 30,000 years. They worked that out through some radiocarbon dating, and they did try to germinate the seeds, but unfortunately the seeds growth aborted, and they think that's because the embryo inside the seed, so inside every plant seed is a tiny, tiny little plant embryo. So it's not a single cell. It's got 2 tiny seed leaves and a radicle, which is a sort of a pre-root, which is waiting to escape. And in some of these seeds, the radicle did emerge, but then the growth aborted. And they think that that's because there was some damage to the growing point, and that then sort of resulted in it failing. But they did, however, they did persist, because these seeds were in fruit and an immature fruit. It meant that the seeds still had some of their placental tissue surrounding them. So if you eat a tomato, some of the jelly-like substance around a tomato seed is essentially placental tissue that supports the seed. And so they were able to, using cell culture techniques, take some of this immature tissue and then put that onto plates, and they were able to regenerate that into callus. And then using various molecular biology techniques, you can take that sort of callus, which is undifferentiated cells, which are just sort of growing into this amorphous blob. And you can use knowledge of how plants differentiate, and you can provide some of the plant growth regulators to those callus, and you can induce shoots. And they did. They got shoot induction, and then they could take those shoots and they could then induce roots from them. And so you got a plant came back from it. And then those plants are then taken out of tissue culture and grown on, and then they were able to produce viable seeds from them as well. It didn't go seed to seed in that sort of traditional way that we think about how we go through a plant generation, but they were able to get living material from that plant and able to get it back to seed such that it was going seed to seed.
One of the initiatives that we are working on with a view to conservation and preservation is to have seed banks where we will store seeds so that in the future, if something terrible did happen, we could bring plants back more easily than the technique you just described.
45% of flowering plant species are likely to be threatened with extinction. And so Millennium Seed Bank is really it's part of that resilience strategy that we're building in order to make sure that should something happen, we're able to get these species back. But there's another seed bank in Spitsbergen in Norway called the Svalbard Global Seed Vault. And the really cool thing here is that they're using the permafrost in order to buffer the seed stored in that bank. So the Svalbard bank stores cultivated plants. So it's more about our crop species, but they store them in vaults buried deep into the permafrost. The idea being that if there's ever any power failures, that the permafrost will maintain the temperature.
Sounds like a very sensible initiative, especially with energy costs being what they are at the moment. Jem Bromley at Churchill College, Cambridge there. Well, finally, we are off to Svalbard that Jem was just referencing over in Norway, but not because of plants, but because trapped in the snow and ice there are microbes, both bacteria and viruses. And like some of the plants that Jem was just telling us about, these are great survivors too. Understanding how they persist there, what they're doing, and how they might also contribute to phenomena like climate change and even antibiotic resistance is the preserve of the University of Aberystwyth's Arwyn Edwards, who's taken fieldwork and Zoom calls, as it turns out, to a whole new level.
Right now, I'm in Ny-Ålesund on Svalbard, which is an archipelago of islands. If you could imagine halfway between the North Pole and the top of Norway, that's where I'm sat at the moment. And I'm probably the northernmost user of a video conferencing app right now.
And what are you doing there?
Leading a research project looking at what's alive in Earth's glacial ice. So 70% of the fresh water on this planet is buried as glacier ice. So in Greenland, Antarctica, and here in Svalbard. And we've come here to Svalbard because we can access lots of different types of glaciers to study what's actually living in the ice.
When you say living, are you talking sort of microorganisms that are alive, growing, proliferating, or are you saying things that are in the ice and they may not be active, but they have the potential to be?
Both really, because for a human or an animal or a plant, you can fairly quickly figure out whether something is alive or dead. For a human, do you have a pulse? Do you have a heart rate? Do you have blood pressure and so on? For a microbe, it's a lot harder to do that because there is no clear dividing line between alive and dead for a microbe. There's kind of a conveyor belt between life and death. And we only know that microbe is dead when it can no longer recover from being really, really damaged on the pathway to death. So there's kind of a spectrum of alive, growing, reproducing through to maybe just able to copy their DNA or avoid damage to their DNA or to express a few genes or to keep some biochemical process just slowly ticking over. And of course, things which are plainly dead as well. So it's a spectrum rather than a very binary kind of situation.
Does it actually have to even be alive or have the prospect to be alive for a microbe to be interesting nonetheless? Because they may nevertheless be almost like a sarcophagus for genetic material, and even though something is not going to ever be viable again itself, it could spew out its DNA, and other viable life forms, microbes, could pick that up and use it, couldn't they?
Microbes love swapping genes from one organism to another. They do that for several reasons. One is to confuse us as microbiologists and make our lives really difficult. The other is because it gives them an advantage in rapidly changing environments or very stressful environments. So when thinking about the vast number of microbes that are trapped in the ice, some of them may be dead, but their DNA might survive. And that DNA may become accessible to other organisms then when those microbes are melted out of the ice. And it's a hypothesis that's been thought about for over about 20 years in my field, and it's been called the genome recycling hypothesis. So we're interested in what those genes are because they could get transferred into modern day organisms and change how they respond to the massive changes that are going on right around me right now here in the Arctic.
How are you studying the bugs that are there, viable or not, in the ice that you've got access to?
Our work here is in three parts, essentially. So the first part is, like today, we've been out on snowmobiles, traveling over a number of different glaciers, spotting where we need to come back to and collect ice cores. And so we collect ice cores from the surface of the glacier, and then we bring them back to the lab here, and then cleanly, as sterile as possible to avoid contamination, melt them very gently, and you can hear the kind of tinkling and fizzing of the gases that are trapped in the ice for thousands of years coming out, when you've got a room full of melting ice cores, and then filtering the bacteria that are in the ice onto a sterile filter paper. And then we extract the DNA from that and use lots of DNA sequencing then to understand what's present in there and what kind of genes do they have.
So these microbes might have been inactive and locked in that ice for millennia.
Yes, so some of them will have been inactive and just frozen in time, essentially. But what I find really intriguing is the ones that may be alive and may be dividing at very, very slow rates, or not even dividing at all, but just preserving the integrity of their DNA or keeping their sort of biochemical processes just about ticking over. And, as microbiologists, we're often used to defining success as what's growing the fastest. But in this environment, it might be the things that are growing the slowest that are the most successful. So we're keen to understand a little bit more about those very blurry boundaries between not being alive and being alive. And that's very curious to consider when you're thinking about life at a glacial pace.
Literally. How do you know, though, that the microbes you're recovering have always been there and they didn't creep in through tiny cracks and fissures or even get onto the ice when you recovered it?
We know that they haven't crept in because the conditions that have led to the formation of ice in this environment. The ice itself, believe it or not, there can be temperate or warm ice and there can be cold ice in technical terms. This is cold ice, so there's no liquid water flowing around within that ice, and there hasn't been any liquid water in contact with it in appreciable amounts for hundreds or thousands of years since it was formed, essentially. So we know that things haven't been able to sneak in from the surface very easily.
And what sorts of microbes are you finding? What lives in ice?
The amazing thing is that there's thousands and thousands of things that live in ice and on ice as well. So I think of a glacier as a sandwich, essentially. So on the top slice of bread, at the surface of the glacier, there's actually a load of things living and growing there in the summer that are very, very happy when there's 24-hour daylight and melt going on. So you get blooms of algae and you get this weird stuff called cryoconite, which is my life's work. But then at the bed of the glacier, you have perennial darkness and cold, and there are things producing methane down there, which is very relevant for climate change. But then in between those two slices of bread is where you've got the filling, and there's a large volume of things. So our best guess is that there's tens of thousands of different species of microbe present in any given glacier, and different glaciers will have different mixtures and different kinds of microbes as well. So when thinking about 200,000 different glaciers across the world, that's a lot of different kinds of microbes and a load of different kinds of microbes in each one.
Are these all what we would dub environmental organisms, the kinds of things you find out there in the natural world? Or are any of them would-be pathogens, the kinds of things that would have come from us or animals and been left behind and now got locked in the ice?
This is a really popular question. To live and survive in a glacier requires a very particular set of skills for a microbe. To live and potentially make a host sick requires a different set of skills. And essentially, you can do either, but you can't really do both. So there's no evidence that there's anything particularly pathogenic to be found in any of Earth's glaciers. We find antibiotic resistance genes quite commonly in some of these environments, but that's because antibiotic resistance is a natural phenomenon. It's just that we've invented antibiotics and it's selected for the organisms that are good at breaking down antibiotics. But in terms of things that can make people sick or animals sick, there's very little evidence for that. And the most fundamental reason there's no evidence for it is that I've been doing this for 20 years and I haven't even caught as much of as a sniffle while doing it.
It's because there's no one else up there with you to give you a sniffle. That's why. But you make an important point, which is sniffles are mainly viral infections. Are you just focusing on boring bacteria? Sorry, I'm being facetious. What about the viruses? Because there are viruses that attack bacteria. Are you seeing any of those, or do you plan to look at those? And are there other viruses that might be up there too?
Yeah, that's a good point. So I handle the boring bacteria because I'm married to a glacier virologist. So we've decided that we shouldn't have professional arguments over who works on what. But yes, to talk about her science a little bit, there are loads and loads of viruses that infect bacteria, bacteriophages, to be found in this environment. It's a very, very tough environment to be a bacterium, not just because it's cold and very stressful for the kind of physical environment, but there are loads of viruses coming after each and every bacterium. So in the general environment, you might find 10 viruses for every bacterium. On glaciers, you might find 40 or 50 viruses for every bacterium. So they are some of the most challenging things that bacteria have to encounter. For human pathogenic viruses, with the exception of smallpox or polio, you're generally talking about enveloped RNA viruses. So that's viruses that have this lipid layer, this fat layer outside them, and have a genome containing RNA. And the most, sort of serious that we've seen has been the 1918 influenza pandemic. And just, I was going to say just down the road here, but a short 20-minute flight from here is Longyearbyen, which is home to a number of 1918 influenza victims who were exhumed in the 1990s. And the researchers looking in the 1990s had much the same kind of technology that we use today for detecting these RNA viruses. And they found absolutely nothing. And that's simply because those corpses had been pushed up into the surface layer of the soil that thaws and freezes every year. So much like you might destroy viruses in the laboratory by freezing and thawing them repeatedly, that was going on naturally. So if there are deadly viruses down there, chances are they're that particular kind of virus, and the physical conditions quickly inactivate them.
Absolutely fascinating. Arwyn Edwards there from the University of Aberystwyth. So there we have it, from mammoths through to microbes, the amazing natural archive that is sequestered in ice and waiting to be brought back to life. Talking of waiting, we will see you on Friday when we're going to be back with a look at this week's top science news stories, and hopefully you also survived the clock changes okay. I've only turned up to one meeting late so far. But I posed that question because we thought this was an ideal opportunity to look at the science of time, including how Greenwich Mean Time, GMT, came about, and also why time flies when you're having fun, like listening to this programme. That is going to come up next week. Meanwhile, we'll also have our regular updates on social media, and if you'd like to support the work we do here at The Naked Scientist, we'd be very grateful if you headed over to nakedscientist.com/donate. Your donations really matter, and we really appreciate your kind support. I'm Chris Smith, thanks for listening, and from all of us here at The Naked Scientist team, until next time, goodbye.